Dr. Chuck the Science Schmuck. Synchronize. So, this may be the first video I'm posting coming back, but I doubt it will be. So I need to explain why that wall is pink. But I do need to explain that part of what I've been doing recently is before I can get my full laboratory operational, I've been going through and redoing older projects that I've done previously. One of the ones that never really did so well was the Winogradsky column. Which, would it be Winogradsky? I think it's Russian, but Russians don't have a W, so there's no W sound. It would just be V, but then it sounds like vinegar, which is what, you drink when you want to drink the pickle juice. And that's really, why is this so sticky? Like, it's been in storage for months and it's still sticky. And it's covered in grit. Regardless of that, I did actually film a partial video of going over the Winogradsky column with the intent of building a more advanced one. I can try to search for that video when I filmed it, but it never actually got developed into a complete video. Alright, so here you see that what looked black is in fact a combination of green and black. The black being bacteria that are producing hydrogen sulfide the black being bacteria that are producing hydrogen sulfide the black being bacteria. So here we see a combination. Sorry, I sneezed. I sneezed everywhere. It's on me. It's on my desk. Oh boy. I can't cover my mouth when one hand is holding a light and the other hand is holding a very thin, slippery cell phone. But here, under this white light, you can see that this is not just black, like it looks like from the main camera. It's actually a combination of black, which is caused by bacteria producing hydrogen sulfide from the magnesium sulfate I applied, and also these interesting green areas, which are green sulfur bacteria, which are utilizing that for photosynthesis. If I move very carefully to the border area, that crusty sort of pink stuff that you can kind of see is not in fact on the outside of the plastic. That's inside. Those are bacterial colonies. Oh, in fact, under this light they look very white, but they're actually a dramatic red color. Maybe retracting the light or changing the angle. Yeah, those are purple sulfur bacteria, metabolizing the sulfur in different ways, coexisting in this part of the column with the other bacteria. And then reaching a point where at the top you actually have photosynthetic bacteria. And it's much harder to film this on the glass one, mainly because the glass is much more reflective. But you can roughly see at the bottom that it's developing the green sulfur and purple sulfur bacteria, various colors throughout the surface of the material, and then finally photosynthetic algae on the top. And right there, if you focus on that, is you. That's what you look like on my camera stand, staring back at me. That's what I see when I'm filming. Smells really weird in here already, and it's just gonna get stinkier. Well, I figured that since I'm already here, I might as well very briefly review what I'm actually trying to do. This is a vase. It's what I'll be building the new ones in. Far less impressive than a big tube, but I think it'll be easier to visualize. Weirdly, they had convex and concave vases. I stood there in the Walmart 25 minutes trying to figure out whether I should use this or the other one. I eventually decided on this one. I think it'll be easier to visualize. If you think of this as a container filled up to here with dirt, the idea of the Winogradsky column is to visualize anaerobic bacterial communities that form on a gradient of sulfur in light, because some photosynthetic bacteria don't bother to use carbon dioxide as we think of plants and cyanobacteria. They use hydrogen sulfide instead as a source of energy. So the idea is that bacteria in the low part create sulfur by digesting proteins and such, and it forms a gradient upward. You have photosynthetic bacteria forming that cycle, and then carbon is cycled up in the top where there is some oxygen access and photosynthesis. My original idea with the other one had been to create one that is so big that it can actually develop a landscape of living plants and animals on the top of it. The problem was, ultimately, you cannot actually build that in this much space. It's too small. An aquarium? Maybe. Maybe I'll revisit it in the future. These will have the advantage of being a potential source for bacteria once they've been purified. Now, the basal layer of this needs to contain two primary features, if I'm recalling correctly. I'm mostly doing this from memory, so the theory may be a little wrong, but when has not knowing the theory behind what I'm doing ever stopped me? It needs to contain a carbon source, which will feed some of the bacteria producing carbon dioxide and sort of generating that process, which in the past I used paper, and it also needs to contain a sulfur source, which would be protein. The protein will be digested and release sulfur. So I have, firstly, the carbon source. Previously I used ground up paper. Did not taste good when I tried to eat it. In this case I have this. Doesn't smell as good as you'd think it would, but it doesn't smell bad. This is depleted coffee grounds. If you're familiar with the way I make coffee, I fill up a pot full of hot water, pour in coffee grounds, let it sink to the bottom, top becomes coffee. Once I've drank that coffee, I fill more water, add more coffee grounds, sinks to the bottom. Over time you get a layer about this thick, it takes probably 3 or 4 weeks to develop it, of just used 3-week-old coffee grounds, which makes the coffee taste real good, but eventually your pot is just full of grounds, and there's no more coffee. So I have to empty it periodically, and usually these get recycled somehow into dirt. In this case, I'm going to use them as a carbon source for the Winogradsky column. You may say, well, what is in coffee that is, nutritive? And I say, have you ever left this stuff out? It does go and it ferments, it gets moldy. About how much I need, I'm not sure. We've got that. Second, I need to add a source of sulfur or protein, of which I have eggs. Now, due to the way the economy is running, eggs are very, very expensive right now. So I was careful. I went through all the eggs in the store, and I was sure to buy a container in which the eggs were already cracked. These eggs are broken. Normally, I would eat the broken eggs because I don't care. I've lost most of my sense of taste because I erased my taste buds with Listerine, like when I was 14. Despite that, I figure I can give these eggs a chance at redemption by becoming part of my science experiment. Give it a dead egg redemption. So, weirdly hard to crack for already being cracked. Yeah, they're a little cloudy in there. Might as well add the shells, too. Calcium. It feels very unpleasant. Alright. Egg coffee ground mixture. I'm very sticky. Now, there is another source that occurred to me, unfortunately, after I bought the eggs, that can contribute to this mixture with a combination of sulfur and protein. I have this still. I don't entirely know why. What is it? Well, let's look at the ingredients. Ingredients. Gelatin. Gelatin is basically cartilage that has been liquefied and denatured from the bones of dead animals, or hooves, potentially. I mean, hooves have a lot of it, too. I used this previously to produce a 2.5-gallon aquarium filled with fertilizer-enhanced Jell-O, so that I could try to grow seeds in it and visualize the root system. What I did not figure is that unlike agar, which is a carbohydrate that most organisms on land can't degrade, gelatin is made of pure protein. And most bacteria, as it turns out, just go right through it. The whole thing re-liquefied and became so horrendously smelly. And then I poured it down the sink and it clogged my sink because it was a giant blob of rotting Jell-O. So I figure it's perfect for this application where it's going to rot in a controlled manner. I added 2 eggs. Let's add 2 packages. Of pure powdered protein. Actually, is it pure protein? Let's see, there's... Well, each pouch contains 1.8 grams, which is the serving size, contains 5 calories and 2 grams of protein. Huh. So it actually contains more protein than the total mass that it is. It does not smell good right now. It smells kind of like dead farm animals, which is what it is technically, but I mean, it's already... no, I'd say it more smells like the waft of an old museum. Like if you were walking through an old museum building, it's like, ew, why does it kind of smell not bad, just old and ugh. So, that is the soup of nutrient source for the columns. Now I need to add bacteria. Where do you get bacteria? Mud. Fortunately, as I'm apt to do, I found a conveniently located drainage pond, wandered out into it, and shoveled up some mud. It doesn't smell that bad. That is oh, there's the smell. Pretty active mud. Oh. You'll know the mud is good when it smells like rotten eggs. And it most certainly does. When it's moving a little bit. Oh well. What always surprises me is that although the mud is opaque, it contains photosynthetic bacteria that can't be exposed to air. So light is getting through a substantial portion of the opaque mud to power bacterial life. Now, the mud itself... Hey, there's a rock in here. Can't have that. The mud itself consists primarily of organic matter and silica. So the key there is getting it real well mixed in with the slosh. I'm trying to get the chunks out too. Like there's a lot of weird rocks because it's a drainage ditch, so they put in quartz in the bottom to help it drain. Weirdly, they're all like the same quartz. If I find a human tooth in here, I am going to be very annoyed. And there is roughly the finished product. That is a mash of decay and... What is that? I don't know. That's the final product. It is a mash of dirt, bacteria, and bacterial sources. I must now place it in jar. Alright, there's the first one filled with fresh water. You can't see it because it's super tiny and dim, but I've marked it with an F for fresh water. It's just water from the sink. I did notice that the sand decreased in volume quite a bit. That's because sand is mostly air. As it gets wet, it kind of collapses. At the end, I will make up the difference in sand levels so that it's closer to what I want. Here I have a bag of Instant Ocean, and here I have a pot of water and some extra water in a Dr. Thunder bottle. Instant Ocean is synthetic sea salt and synthetic salt, and what does that mean? It means it's produced in a laboratory from various chemicals to mimic the chemical constituents of the ocean. It's what you use in saltwater aquariums, which that saltwater aquarium I set up is mostly dead. There will probably be a video of me resurrecting it in the future once I can find which state I left it in. As you can see, it is salt. Well, I don't know entirely what I was expecting. It tastes like salt. And it's not salt, exactly. It's not table salt, which table salt is primarily sodium chloride, and weirdly a lot of cyanide. Iron cyanide, though, so it's non-toxic. Gotta mix it. The weird thing about bacteria is that a lot of times they don't follow the rules that you would expect logically from observing, fish and other organisms, and that salinity to, like, fish or humans is a huge deal because there's complicated things like organs and kidneys and other things that need to be able to work at osmotic pressure. Bacteria, though, they're single-celled and really weirdly hardy, so some bacteria don't even care exactly the level of salinity they're in. It's not hugely strict. It's almost terrifying how durable bacteria can be just on a population basis. Like that mud, right? The bacteria in there that I'm looking for are anaerobic, obligate anaerobic, like Clostridia. They can't survive in the slightest exposure to oxygen. Oxygen is extremely toxic, right? It produces dangerous free radicals in cellular metabolism that our bodies can deal with because we produce, superoxide dismutase and catalase and stuff to break that down. They don't. If they're exposed to oxygen even slightly and absorb it, it poisons them. Oxygen is extremely deadly. And yet they're still in there, even though I just had that mud out. Why? Because bacteria are so tiny. 2 tiny grains of sand, silica, clay clinging together contain thousands of bacteria between them. And it's like, oh, that tiny colony out of the billions of bacteria I put in, if 1 million survived in 10 days, I mean, that's like less than, that's 0.1%, right? 1 million of 1 billion? No, it's 10%. No, 10 million, 100 million, 1,000 million. It's 0.1%. So if 0.1% survive, that's tiny, but it's still 1 million bacteria, and in 3 days they'll be back to normal. That's why when you see stuff that says, oh, it kills 99.99% of bacteria, that means nothing. That 0.001% you've left alive will be back to full strength in a matter of a couple hours. Maybe not hours for most bacteria. I mean, it would have to be an ideal culture situation, but it is still billions of bacteria survive if you kill 99.99% just because there's so many. So I've mixed some salt water and I have a hydrometer. So as you can see, this water is very salty. 1, because there is literally salt floating in the pot. And 2, the needle is pointing all the way up. That means this is very hypersaline water. This one gets to be the hypersaline container. That salt water is just going straight in. So that is a portion of brine, which is what the word for very salty water. This, contrary to its label, is not Dr. Thunder, because who needs to label chemicals properly? It's fresh water. Allow me to add some to see what happens when the two combine. Okay, the result is that the water is slightly saline. It is not ocean salinity, which is, you can't really see the color anymore because the thing's so old, but it starts at a specific gravity of 1.020. So if I add this whole thing into there, we might come close. I'm sticky from when this was full of soda. Now everything is salty. Worse, I now need to go to the bathroom. Huh, I didn't even do math. It came out on its own. The needle is now right about in the middle. This is not fully dissolved, so this would not be prudent to use for an actual saltwater aquarium because some of the specific nutrient, like hard to dissolve salts, have not dissolved. But for just maintaining salinity and bacteria, it should be fine. The needle is approximately where the ocean would be. Granted, it will drop slightly just because there's inherent moisture, but you also have to consider diffusion through the system and how fast does the salt and water osmotically shift to draw similar water concentrations. So it's close enough, basically. I say basically a lot because salt water is very basic. That's an interesting feature of this, that while sodium chloride doesn't really have a pH distinct to itself, this is also, in addition to being salty, very alkaline. Tastes like the ocean, except without all the, lifeguards. This salt's actually, you can tell there's water in the bag, and you say, how did water get in there? It's hydroscopic. Pulls water out of the air, and then drips down this super brine out of itself. This stuff probably is expired, and I need to get new salt. So I have fresh, salty, and very, very salty. Oh, there's an air bubble. Now, as I mentioned previously, I need to add sand, and there you have it. These three are set up with their different salinities, with their sand, and with their water. That was a little low, so I'll top it off. But the idea now is to just put these on a bright windowsill, which I have over here, and let them sit, slowly rotating them and ensuring that the water stays roughly the same as where it is. The thing about salinity is that I don't need to refill it with salt water. As it evaporates, the salt stays in, so I just add fresh water to keep it up. Ideally, they will begin to develop, one, decay reactions here, producing carbon dioxide and hydrogen sulfide, and then a spread of bacteria that will grow across the inside surface of the jar through this sand that are utilizing sunlight as energy. This time, I intend to actually show you the outcome, so this video is actually going to go into stasis for a really long time, probably about 2 or 3 months while these grow, and then I will finish it once they've actually started. And that's why it's one of the first videos, so that I have time to let this grow so I can post it in the future. So that time will pass very slowly for me, but it'll be near instantaneous for you.